Hello, hello, hello. Back to finish up our assay notes. So 2.3 is all about RNA and transcription. We'll get transcription in just a few minutes here. But transcription is the process of going from the DNA code to RNA. Before we get to all that, though, here is one eukaryotic chromosome. So we're looking at one eukaryotic chromosome. It kind of looks like this. You can see it under a microscope and straighten it out. So this is one long length of DNA. That's what a chromosome is. That gigantic pile of DNA, those A's, T's, C's, and G's, they're all going to kind of coil and supercoil and make a structure that looks like this right here, which is a chromosome. Humans have 46 of these. So we have 46 of these stretches of DNA that we call chromosome. Usually they're somewhere, now they vary in length. Some are long, some are short, but generally they have between 50 million and 300 million. Oops, not 30 million. Got a zero there. 50 million and 300 million nucleotides. So each of your chromosomes has between 50 million and 300 million A's, T's, C's, and G's. So there are a lot of letters in each one of these strands. Now, again, this is different than the eukaryotic chromosome. We mentioned that last week. And the eukaryotic chromosome, again, we have a beginning and we have an end. It's a stretch of DNA. Prokaryotic chromosomes are circular. So there is no real beginning or end, but with ours, there is. All right, so why are we talking about this? We just finished our DNA part. Again, here's the long stretch of DNA. Here's where we're getting at this today. This stretch right here. Give it a little different color there. This stretch right here is one gene. When we get to transcription by the end of today, we're going to see that we don't transcribe the entire chromosome. We don't start at the beginning, we don't end at the end. We just do a very small segment of the chromosome, generally about what we call one gene. Don't worry about genetics, don't worry about any of that. We'll get to that. We have a whole couple of chapters on that later in the year, but for right now, we're only turning a small section of this into RNA. We're not copying the entire chromosome. One gene is just a few thousand nucleotides. So just a very small fraction of the 50 million to 300 million nucleotides found in the entire chromosome. We're just doing a few thousand. So not the whole chromosome at all. Now also keep in mind oops. So also keep in mind that there's going to be several hundred genes on each chromosome. It's not just one gene per chromosome. There's going to be several hundred, up to I think it's 800 genes on some of your chromosomes. So the same gene, the gene for eye color and the gene for height might be a little bit further down and a different gene down here. And depending on what that cell is trying to transcribe, trying to turn into protein, it's going to read that particular gene and it's going to turn it into mRNA first. And that's what we're getting to today. We'll worry about the protein translation a little bit later. All right, so again, take-home message from drawing our chromosome here is that when we transcribe into RNA, we only do a small little piece of it, not the entire length. All right. right, let's get into RNA just a little bit. Just like DNA, RNA is made up of repeating monomer units called nucleotide. And just like DNA, you remember this from last week, the nucleotide has 3 parts. One part is a phosphate. This should look familiar. We also have a 5-carbon sugar. Now, different than DNA, RNA, the sugar is called ribose. So there's the R in RNA. If you guys remember DNA, we used deoxyribose, and that was the D in DNA. Well, the R in RNA is from ribose, which is the 5-carbon sugar used in RNA. And again, just like with DNA, we also have a nitrogenous base as part of every single nucleotide. And hey, keeping those similarities rolling, there are 4 different nitrogenous bases, A, C, G, and then here's the difference, a U. There's no T in RNA, it's a U, and the U stands for uracil. Uracil. So uracil replaces thymine in RNA. So the thymines are in DNA, we use uracil for RNA. A couple of other noteworthy differences and similarities. So just like DNA, RNA has a sugar phosphate backbone. So these nucleotides are going to be connected, phosphate, sugar, phosphate, sugar, phosphate, sugar, just like DNA. Different than DNA, RNA, the bases, that they do pair up. They do and they will pair up, but they don't stay paired up. So different than DNA, RNA is usually single-stranded. Again, the base pairing rules still apply. We still have them. C always pairs with G, G always pairs with C, T would always pair with A, and A always pairs with U. Remember, U, it's RNA, it's not DNA. So there's a couple more similarities and a couple differences. Another difference, so flipping the page here, need a little bit more room. Another difference is that there's 3 types of RNA. Actually, there's more than 3 types. There's 3 main types that we're going to focus on for this class, and that's different than DNA. With DNA, it is what it is. DNA is DNA is DNA. There's not different, really different types of it. It just has its job and it does its job. Whereas again, RNA, we have 3 different types. The first type that we'll work with is called messenger RNA. And you're almost always going to be seeing it written as just mRNA, where the M stands for messenger. Make sure you guys know messenger RNA. Messenger RNA kind of looks like this here. And it's single-stranded. There's our sugar phosphate backbone, and then the base is coming off of it, but they don't base pair, they don't stay base paired with another strand, so we don't have that double strand that we're so used to seeing in DNA. The messenger RNA carries the DNA message, so you can see why it's called messenger RNA. Oops. Went ahead of myself up here. Another type of RNA is called ribosomal RNA or rRNA. And the R standing for ribosomal. And as you might have guessed, ribosomal RNA makes up a ribosome. And this is actually fairly accurate. I'm not a great artist, but this is a fairly accurate representation of what a ribosome looks like. It almost kind of looks like a hamburger bun. You got the top part's a little bit bigger than the bottom part, kind of holding on to it there, your hamburger. So there's our ribosome. And as you might have guessed, it makes up a ribosome. Now there's going to be proteins mixed in there as well, but a lot of rRNA is mixed in there as well. So there's a ribosome. And then finally, the third type of RNA that we'll work with a lot is transfer RNA. Perhaps you guessed what the abbreviation for transfer RNA is. I bet you did. We call transfer RNA tRNA. And transfer RNA transfers the genetic code into protein. And it kind of looks like this, little loop-de-loo. We'll talk more about this next chapter. At the top here, I'm going to put a little AA. Not important for this particular unit, but I want you guys to be aware that it's there. When we get to our next unit, we're going to talk a lot more about that. The little AA stands for amino acid. Again, we'll talk a lot more about that next chapter. For right now, I just want you to know kind of that it's there. And ultimately, guys, we're going to work with messenger RNA a little bit more here this next section. But these two, rRNA and tRNA, don't sweat them. Don't sweat them. All you need to know is exactly what you see. There's rRNA, and it makes up the ribosome. And there's transfer RNA, and that's what transfers the genetic code into protein. Outside of that, we'll work more with these two, especially next unit. Don't sweat too much about them. So our next section here is RNA transcription. Again, just so we're all clear of what that word means, transcription, we're going from DNA to RNA. That's what the word transcription is, and that's what we're looking at in this particular chapter, or this particular section. All right, so drawing our drawings here. Oops, I came in a little too far with that one. Okay, so hopefully we're kind of recognizing this here. Maybe that helps. There are definitely days I wish I was a better artist, so I apologize. But hopefully you're getting that general picture here. Oops, these are going to be closed. They're not open. So just the big bubble here is open. And this one's back to the DNA being together. And then here's another bubble. We'll talk about these bubbles in just a second here. So it's open. There's the open nucleotides, the exposed nucleotides, and then closed DNA again. All right, so if we're kind of recognizing what we're seeing here. Now I'm gonna skip a few lines because I'm gonna add a few more drawings into this area here. I'm gonna take a couple notes then, and we're gonna start. Let's start right in this general area here. So we're gonna call this the Tata box. So on our first stage, and I'm skipping a couple lines so I can continue drawing. But in our first stage of transcription, of copying the DNA code into RNA, our first stage is initiation. And that has to do with the promoter sequence, which, as we've drawn it, is the Tata box, which is a lot of humans and a lot of other eukaryotes as well. The promoter sequence is a series of a lot of T's and A's, T and A and T and A and T, and therefore it's named the Tata box. So a whole bunch of nucleotides that go T and A and T and A and T and A. The Tata box is our promoter sequence. The promoter sequence has three things it needs to be able to do. Number one, it attracts RNA polymerase. Our old friend RNA polymerase. You guys remember we made him a square, I believe. Last section, we talked about his roles in DNA replication. So here's our square, and he's going to come and kind of land. It's like a runway for him to land on the DNA. And he's attracted there by the promoter sequence, which in eukaryotes is usually the Tata box. Second thing, the Tata box signifies to RNA polymerase which strand needs to be transcribed. So this is kind of an important concept here. We don't copy both strands of DNA. Remember, when we did DNA replication, we had to copy, we opened and then copied both strands. I'm kind of going over just one strand. I don't want to get confused later on. So I'm going over just one of the strands with the blue color so we can see it just a little bit better and it stands out to us. We're only going to copy one strand. We don't have to copy them both. We don't have to worry about going leading strand, lagging strand. We just go in one direction. And which direction that is, it's the Tata box. His job to tell RNA polymerase where that is. And then finally, also, as you might imagine, it's a promoter sequence. So it's going to tell RNA polymerase, hey, this is where you begin. So it's going to come in, land on the Tata box, and you're going to start transcribing. I'm going to go ahead and draw another RNA polymerase here. I'm not necessarily doing a time lapse. In DNA, we did some time lapses and said, okay, fast forward 5 seconds. When our cells decide it's time to make a protein, they get to work. They are going to copy the heck out of this code. They're going to make hundreds, maybe even thousands, maybe more than that even, of mRNAs, of copies of this gene. So they're going to call over hundreds of RNA polymerases and just get them going and get them going and get them going and get them going. And they're going to make tons of copies of this and then tons and tons of proteins. It's not like a once over thing. So here is our polymerase, and he's adding nucleotides here to a growing strand. And how did the strand come off? So see how the strand kind of deconnects from the DNA? He's only bound for a little bit. He's not bound very long. Then he kind of comes loose. And this, we just took notes on him. This is our mRNA strand. And he gets built, he gets base paired. Whenever RNA polymerase reads a T, he adds an A. Whenever he reads G, he adds a C. Whenever he reads a C, he adds a G. And whenever he reads an A, he adds in U. Hopefully, you guys all said U. Again, RNA does not have T, so never put a T into RNA. So there's our RNA sequence. Again, he's only base paired for a little bit. He doesn't stay double-stranded for very long. It's going to kind of peel out of there. All right, now I had hoped to get this done just, on one page. Unfortunately, I'm out of room. I'm going to flip, and we're just going to have to kind of go back and forth here on pages for a little bit. So the second stage of transcription is elongation. And during elongation, RNA polymerase first, let's use the word unzipped. I think that's the word we used before. So first, he unzips the DNA strand. Second, he builds an mRNA strand using the base pairing rules, which we just talked about and you guys are familiar with. Kind of a side note then. The transcription level is going to be about 10 to 20 nucleotides long. And just so we're all aware, we've kind of got this bump you kind of notice here. This is a transcription level. Got another one over here. And these transcription bubbles are about 10 to 20 nucleotides long. So the RNA polymerase is unzipping the DNA, he's opening it up, he's copying the mRNA. Only about 10 to 20 nucleotides are exposed at a time. And then after the RNA polymerase has passed that segment of DNA, the DNA comes back together. A lot easier than DNA replication. Much, much easier. Remember, we had all the different proteins. We had to have helicase unzip the DNA. Had to have the SSBPs hold it open. Had to get started with a primer. This guy's a primer as well, primase as well. This is a much simpler process. RNA polymerase pretty much handles everything. Just opens up the DNA, makes a strand, and the DNA just kind of closes up behind it. So it's a much, much simpler experience, RNA transcription. All right, we have one more stage to talk about then. And that is termination. It's pretty much exactly what it sounds like. At the end of the gene, so I'm gonna draw one more RNA polymerase down here. Again, he's adding his nucleotides, and then he's got a tail of mRNA coming up. I'm gonna make this tail a little bit longer. You guys notice that little artistic effect there? His tail's just a little bit longer because he's been at it for a little longer time. He started here just like this guy, just like this is about to, but he's gone all through this area. Whereas mRNA here is not as long because this RNA polymerase is only a little bit into it. He only just started. So his tail's a little bit longer. He's got a longer mRNA strand, and he's about to get to the end. So right here is going to be another sequence of DNA that we are going to call the termination sequence, which is exactly as it sounds like. Now, while we're here, I'm going to add a few things. I noticed I forgot to add. So if you remember, I colored the strands a little different color here to really make them stand out. Well, that blue strand, the one that's being copied, is the template strand. Whereas the one that's not being copied, interestingly enough, this is called the coding strand. And if you think about it, it should make sense. All right, let's go ahead and finish up our drawing then. At the termination sequence, let's add one more RNA polymerase, but he is falling off. Got to the termination sequence, and then he came off. Now I'm going to add a mRNA strand, a completed mRNA strand down here at the bottom with him. He fell off as well. So there's a completed mRNA strand. It came off with this RNA polymerase. He got to the end and he was done. So they all came apart. Basically what our notes are going to be. So at the end of the gene, remember, we didn't transcribe the entire length of DNA. We're only doing one particular gene. So when we get to the end of the gene, we have the terminator sequence. RNA polymerase falls off. Oops. And that completed mRNA strand, the copy of the DNA, heads to what we call editing. Hey, that wraps up what we have to work with for this chapter. When we pick up again next chapter, we're going to pick up right there, the RNA editing. Quick shot at that drawing. Here is the idea of RNA transcription, going from the DNA code to the RNA code. We have a promoter sequence that calls over the RNA polymerase. Polymerase is only going to transcribe one strand of DNA, doesn't need to do both. There's going to be hundreds of these RNA polymerases all landing and transcribing the same gene. Once they get done with the gene, they reach the termination sequence, they fall off, and their completed mRNA strand is going to float away and go to what we call editing. And that's what we're going to pick up next time. All right, guys. Well, as usual, it's been a blast. I'll see you next time.